These are notes on evidence for the theory of evolution, and you can find additional reading on pages 107 to 109 in your text. And then there's also a great taxonomy chart, how we name things, how we categorize things, on pages 76 to 77. So if you remember from the start of the year, evolution is a theory, and theories in science are accepted by the scientific community and have overwhelming evidence supporting them. And I'm not going to overwhelm you with evidence tonight, but I'll at least kind of show you the progression of how we understood evolution and the evidence we had in the beginning and then much later on and then what we have today. So I realize I don't have a slide on Darwin, but I also forget every year to mention Darwin and then realize that there are still some students who don't know who Charles Darwin was all the way through the evolution unit. So Darwin is the scientist that is credited with putting together what we call evolution by natural selection, putting together all of those ideas into a coherent theory, collecting evidence for that theory. I would say, like anything else in science, you know, we talked about Rosalind Franklin with DNA and Watson and Crick getting the what's the word I'm looking for? Accolades for recognition for DNA, even though certainly other scientists like Rosalind Franklin did a lot of work. The same thing with evolution, right? So you can spend years looking at all of the scientific controversies over who should have or should not have really received credit for stuff. But Darwin is the scientist that we credit with formulating the theory of evolution. And he did it, really, he was a naturalist. He sailed all over the world on his ship, the Beagle. He categorized and cataloged life and really looked at how different species seemed evolved to whatever habitats in which they lived. So that's who Darwin is. So now let's get to some evidence. So we have fossil evidence first and foremost, and fossil evidence showed a couple of things. They showed that organisms changed over time. So you could find two fossils that looked very similar, but there were differences. So the idea being, if you've got two fossils that are similar yet different, that one may have changed from an earlier species, so that organisms do in fact change over time. The other thing it showed, which I realize I don't have a bullet point for, is it showed that there were extinctions. So I know that that seems really evident to us now, right? We have endangered species, we have organisms that we know have gone extinct, but that was a really novel idea, that things would go extinct, that everything that's on Earth was not always on Earth, and there was more stuff on Earth that is no longer around. And so fossils really gave us evidence of that. And they can also provide evidence of relative and actual time. So if you look at this cross-section of strata here, strata being the Earth, we have what's called relative dating on this side. And relative dating is simply saying younger and older. So the idea being that the further you dig down, the older the rock layers are. So you could look at something like these shell fossils here, and those are going to be a lot younger than these trilobite fossils here, simply because the shell fossils are closer to the surface of the Earth. But we can also use different strata to look at numerical dating. So you've heard of carbon dating of living organisms. So we use, or let's say biotic organisms that are currently dead, but they were biotic. You can use carbon dating, but carbon dating is actually only helpful, I want to say, to a couple 10,000 years. After that, it's not that helpful. Like when you get into the millions of years, it's definitely not helpful. And it has to do with the half-life of carbon, which you don't have to worry about half-lives now, but it's essentially how quickly elements break down. So if you're using an element that really most of it is gone after, it's all broken down after, say, 50 to 100,000 years, which is long, but when you're looking at millions of years is not, then after that time period it's not helpful. But you can also do what's called radioactive dating, and that's dating of volcanic ash. So you can see it says in here, dating of volcanic ash. And what that is when something has been spewed out of a volcano, we kind of time that as time 0. So we can use radioactive dating and we can trace elements that have much longer half-lives, meaning stay around way longer, like uranium's one. I want to say uranium takes 4.5 million years for half of it to break down, to start dating some of this. So if we know, for example, that this volcanic ash is 520 million years old, and this volcanic ash is 545 million years old, then we can start to date these trilobites and say, you know, they're somewhere around 535 million years old, somewhere in there. So that's fossil evidence. We can also look at anatomical features. And so we're looking at body structure. And there's some anatomical features we can look at, and there's some that we can't look at. So the first kind are analogous structures. Analogous structures, they perform similar function, but they don't have a similar structure. So we've talked about structure and function. You've learned about structure and function since you were in kindergarten. So we understand that structure is how it's built, whereas function is what it does. And analogous structures we cannot use to show kinship. It doesn't mean that they had a similar ancestor simply because they have analogous structures. So analogous structures are like this. A shark has a fin, okay, and a shark is I mean, they say fish, but shark is actually different from a fish. We won't get into why. And then a penguin, which is a bird, and that has this kind of wing-like structure, and really any bird has a wing, we know. And then a dolphin, which is a mammal, has its little flipper here, and all of these help this animal to be streamlined in the water, right? And the penguin is, of course, an aquatic bird. Penguins don't fly. But if you look at the underlying structure, the bone structure, you see that these are really different structures. And so what that tells us is these are analogous to one another, so we can say they do the same thing, but they don't show underlying kinship in this. So we wouldn't look at these and say, oh, a penguin must be really closely related to a shark and a dolphin because they all have these streamlined appendages, streamlined flippers or wings, depending on which one you're looking at. That's analogous. The other one, though, is homologous. Homologous structures are exactly the reverse. They're similar in structure, but they're different in function. And these do give us evidence of a common ancestor, okay? So, for example, if you look at what we call pentadactyl limbs, so penta meaning 5, dactyl meaning appendage-like things, you can see that they all have these 5 appendages, 1, 2, 3, 4, 5 fingers here. The bat here has 1, 2, 3, 4, 5. The whale has 1, 2, 3, 4, and then there's a 5th one down there. The cat has 1, 2, 3, 4, 5. And you see they all have a radius. This is the radius. And they all have an ulna. That's the ulna, and you can see that on all of them. It's nicely color-coded, and they all have these wrist bones. So they all have very similar structures, but the fact that they have different functions leads us to believe they have a common ancestor. And the reason we say that they have a common ancestor is because it would be very unusual for these 4 organisms to develop 4 such different functional body parts all with the same structure. So probably a much more reasonable explanation than just they happen to all have the exact same bone structure is that there was some common ancestor of these 4 organisms, and that structure then changed based on the habitats of the four organisms. So the bat developed more of a wing because it was an airborne creature. The whale developed more of a fin because it's an aquatic creature. We developed hands because we like to point at people with our fingers. But the fact that our hands have the same bone structure as a bat's wings, even though we use them for totally different things, really gives us some evidence that there's got to be some common ancestor there that we then all broke off from. Now, that said, the shark, the penguin, and the dolphin are all animals, and they are all what we would call chordates. So they're all, in fact, the same phylum. And so they do clearly have a common ancestor, but you can't use their fin, wing, and flipper to show that they have that common ancestor. You'd have to use some other type of evidence. And that if we look at the dolphin, which dolphins and whales are incredibly closely related, hopefully that's not news to anybody, so we can have the dolphin here, we would say that based on anatomical features, dolphins and whales are much more closely related to bats, cats, and humans than to sharks and birds. So there's another kind of anatomical feature, and that's called vestigial structures. So vestigial means it's something that had a function in some common ancestor, and you see evidence of it in current organisms, but it really doesn't have a function anymore. So a really great example in humans is the appendix. Other than occasionally blowing up and needing to be taken out, there doesn't really seem to be a function of the appendix in current humans. So you can also see on, so this is a human here, and then this is a snake, and this is a whale. And all three of these have what's called a pelvic girdle here. So ours is in fact our pelvis, our kind of hip region, okay? And ours we use, it's where our hip is connected. It's one of the major bones of our body. You don't want to break your pelvis. I know someone who did that. It's miserable. The whale also has a pelvic girdle, and then they also have, the snake and the whale also have these vestigial hind limb structures. Okay, now snakes have no limbs, and whales have no hind limbs, right? We just saw that its flipper here is its forelimb, but they all have a pelvic girdle, they all have hind limbs. This shows they also have vertebral columns, but that wouldn't be a vestigial structure. But the hind limbs and pelvic girdle in both the whale and the snake are vestigial structures. They're not being used anymore, but they're still slightly there. If you didn't look at a skeleton, you would never know they were there. But they are there, which gives us evidence, you know, why would something have a structure that it's no longer using? Well, it must have used it in the past. And so clearly over time, because these weren't useful structures for the habitats to which these animals were adapting, they got smaller and smaller and smaller. Now what's also crazy about this is this gives us really good evidence that whales originally, and some of you know this, but here's evidence of this, that whales originally were land animals, and they had hind limbs, and they walked around on them, and that at some point they went back into the water, right? We know that all life originated in the water, but that whales were actually land animals that went back into the water. And so I just kind of want you to think about what would cause a land animal to go back into the water after being on land long enough to evolve hind limbs and then go back into the water and lose that trait. So another set of evidence is called embryology. So looking at embryos and noticing that embryos of different species look similar, even though the baby of that species, when it's born, does not look similar. So the two examples that are often given are gill pouches and a bony tail. So like here in a human, you can see they've got this bony tail here. Here they have the gill pouches. And you see the same one in a chicken. And for whatever reason, the chicken and the human are always compared. Although certainly I think the turtle and the human look a lot more similar. And, you know, in humans, those gill pouches later develop into the whole ear, nose, and throat system, and there's something different in a chicken. But at some point in the embryological development, those species look similar. And so again, kind of like homologous structures, if we're seeing similar development patterns, yet they develop into different structures, it's got to be because at some point this was one organism developing into something, and then all of these other organisms branched off. As an interesting aside, it used to be thought that how an organism developed, that like as humans developed, they went through the stages of all the quote lower animals. So, you know, it was thought that humans were sort of the pinnacle of evolution, and so as we developed, we went through all the stages of the other animals in utero. So if a mother had a miscarriage and the embryo maybe came out kind of looking like this, they'd say, oh, this baby got stuck in the turtle stage, say, you know, and they'd say, see, you can see these gill slits here, you can see this tail, that it clearly was stuck in the turtle stage. We now know that that's absolutely not true, but we can use embryos to notice a common ancestor. And then we have DNA evidence. And realistically, the only thing people use these days in science is DNA evidence. Nobody says like, I think these two animals may be related, or these two trees, or whatever. Let's look at their skeletons. Let's look at their skulls. They say, let's look at their DNA and see how closely related their DNA is. So the more similar the DNA, the more closely related the species. And so in the video, we know that if you look at lengths of DNA, right, anything that undergoes cellular respiration has got to have DNA for making mitochondria, for metabolizing sugars. That can't be coincidental that all that DNA is exactly the same. There's got to be some sort of, again, common ancestor. So if we look at these 5 species, we've got human, horse, gorilla, chimpanzee, and zebra. And then here's a sequence of amino acids in the same part of hemoglobin molecules. Okay, so we know hemoglobin is a protein. We know that these amino acids are coded for by DNA. So we can look at amino acids instead of just A's, T's, G's, and C's and say, are we seeing the same patterns in making of the hemoglobin? Now what's interesting is all of these animals make hemoglobin, but it's a slightly different molecule because it's a slightly different protein because they're slightly different amino acids. But go ahead and pause the video and take a second to just see if you can tell based on this which animals are most closely related and which are least closely related. So if you compare them, you can see that, for example, the human here and the chimpanzee here have no differences in this section of their hemoglobin. And so we would say that those two organisms are going to be the most closely related of all of those. Then if you look at the human and gorilla, or the chimpanzee and gorilla, there is one difference between them. So instead of this isoleucine, this is, maybe that's lysine. And so there's one difference. So we would say the human and chimpanzee are more closely related than the human and gorilla or the chimpanzee and gorilla. But those two only have one difference. So those 3 together are more closely related than, say, the horse, which is 2 different from the gorilla. And so 3 different from the chimpanzee and human. But then the horse and the zebra are only off by 1 amino acid. So those two have got to be really closely related compared to the horse and the gorilla, or the horse and the chimpanzee, etc., and so forth. So we can look at DNA just and say, you know, which DNA is closest, because whichever is closest had a more recent common ancestor, meaning chimpanzees and humans broke off from each other after they broke off from gorillas. So there was a gorilla, or there was some common ancestor, and then gorillas went one way and some other organism went the other way, and then chimpanzees and humans broke off from that other organism. And we're going to make trees called phylogenetic trees to better understand this. Then we also have examples. So, in Darwin's time, there was no understanding of the fact that we could see evolution happening right before our eyes. So if you look at, like, say, the evolution of, I don't know, the turtle, we're not going to see anything in the course of our lifetime, right? Evolution is changes in populations, not changes in individuals. And so it takes millions of years. But if you look at something that has a very short lifespan, days, weeks, months, we can start to see evolution happening. So this is 3 different bacterial strains: Staphylococcus, Enterococcus, and Pseudomonas. If you care about the species names, go for it. These are all genus names. And these are all bacteria that harm humans. And then 3 different antibiotics that are used to control those. And if you look at the Staphylococcus, which is often killed off by methicillin, in the 80s, almost no Staphylococcus was resistant to methicillin. Okay, we now call it MRSA, methicillin-resistant Staphylococcus aureus. But over time, as we've used more and more antibiotics, and so, in a sense, it created an environment where being resistant was advantageous to the bacteria, we see a huge increase in antibiotic resistance. Now, these are bacteria that have been around harming humans for hundreds of years, right? They didn't spring up in 1981. But we can really see over the past 30 years a huge increase in resistance because we're creating environments where it's advantageous to be immune to these antibiotics, which is why your doctor will tell you you don't want to take antibiotics if you don't need them, because that encourages more and more bacteria. It creates more and more environments where being immune is a good thing if you're a bacteria. If you're a human, you want to be able to take methicillin or vancomycin and kill off the bacteria. You don't want it to be resistant. All right, so at this point, please go ahead and add any questions you have to the questions part, or ask them in class after we take the notes. Have a good night.